Hello, I'm Brad Zuchero, PhD, Assistant Professor of Neurosurgery at Stanford University and 2019 Beckman Young Investigator. Today I'll be telling you about work in my lab to elucidate new roles of myelin in plasticity, learning, and disease, specifically by building first-in-class genetic tools for perturbing myelin in vivo. The human brain is composed of billions of neurons, but the brain is more than just neurons. In fact, more than half of the cells in the brain are glial cells, non-neuronal cells that I'm showing here on the slide. The microglia, the resident immune cells of the brain in pink, astrocytes, glia with diverse functions in neuronal survival, the formation and elimination of synapses in green, and oligodendrocytes, the myelin-forming glia of the central nervous system in blue. The word glia comes from the Greek word for glue because traditionally neuroanatomists and early neuroscientists thought that glia only played passive supporting roles in the nervous system, or were really just brain glue. But in the last few decades, we've realized that glia have very, very important and exciting roles in development, dynamics, and diseases of the nervous system. My lab focuses on oligodendrocytes, the myelin-forming cells of the central nervous system. Myelin serves as the electrical insulator of the nervous system, similar to this electrical insulation on the copper wires in the circuit. Myelin is essential for the electrical signals to travel fast, as well as to be timed precisely through the nervous system. Myelin has broad disease relevance. It's well known that myelin is directly affected in multiple sclerosis, leading to very profound disability in patients. Myelin also has emerging roles in neurodegenerative diseases like Alzheimer's, as well as broader roles in epilepsy, injury, stroke, and even neuropsychiatric diseases. It was long thought that myelin was just made innately during development and then statically maintained in the adult. However, emerging evidence suggests that myelin actually plays way more dynamic and exciting roles in the adult nervous system than previously imagined. Human neuroimaging studies found that when people learn new motor tasks like juggling or playing the piano, the major changes in their brains are actually in their myelin or in their white matter tracts, suggesting that the remodeling of existing myelin may actually be important for learning and memory. Researchers have found similar things in the brains of mice. A major question for the field is how extensive myelin's role is in learning and memory. However, there's a major barrier that's holding us back. That barrier is that we lack precision tools to perturb myelin in vivo. This is the goal of my Beckman Young Investigator Award. How might we be able to build tools to perturb myelin in vivo in neuron-specific ways? First, we have to think about how an oligodendrocyte actually myelinates a neuron. So during development or in the adult brain, oligodendrocytes extend and retract dynamic processes, their fingertips, that select and then spirally wrap around neurons to form this insulating myelin sheath that's important for rapid nerve signaling. This process requires a very precise and intimate one-to-one cell contact between the oligodendrocyte and the axon, cell-cell contacts that we're just beginning to understand. Our goal is to create tools that can perturb myelination by perturbing that specific cell-cell contact between the neuron and the oligodendrocyte. In our vision, we would create tools that we're calling imagines for inhibitors of myelination around genetically defined neurons, shown here in red, that when expressed on neurons effectively cloak these neurons, making them unable to be myelinated. So you see here in this graphic, the gray neurons running horizontally do not express imagines and are able to be myelinated like normal, whereas the diagonal red imagine-expressing neurons were unable to be myelinated. We're designing these constructs in a modular manner to use DNA sequences for myelin-blocking domains, shown in red, coupled with the green fluorescent protein to tag these proteins, and then axonal targeting sequences to act sort of like zip codes to drag these constructs out to the neuron's axon. We envision that these tools would localize to the surface of the axon, expressing this myelin-blocking domain on the extracellular side that would effectively block the axon from being myelinated by an oligodendrocyte. In designing imagines, we were inspired by Dr. Beckman, who famously said that he did more for science by making instruments available for thousands than what he could have done in the laboratory by himself. In a similar spirit, we're designing imagines for widespread use and adoption by the entire research community. First, we use an AAV virus-based design that allows for rapid prototyping and testing in vivo to accelerate the time from idea to discovery. We're building these tools to allow for plug-and-play modular construct architecture, keeping this all open source to allow for other users to rapidly modify or improve on our designs. Finally, we're freely sharing and widely distributing these to all research groups, making use of AdGene, the nonprofit global plasmid repository. Sometimes science takes us to unexpected places. While prototyping Imagine's constructs, we stumbled upon a protein, known to be linked to disease, that causes oligodendrocytes to undergo a dramatic transformation, unlike anything we expected or ever seen before. This signal causes myelinating oligodendrocytes to turn into something we're calling a transformed oligodendrocyte that is a dramatically different cell state. Transformed oligodendrocytes are characterized by rapid and robust myelin degeneration, dramatic gene expression changes of thousands of genes, as well as the adopting of new functions like phagocytosis, something an oligodendrocyte normally never does, shown here in this video. Thanks to the support from the Arnold and Mabel Beckman Foundation, a high-risk, high-reward project like this, focusing on our discovery of oligodendrocyte transformation, can now become a major research focus in the Zuchero lab, and we're hoping that this will have a major impact on the field in the years to come. In closing, we believe that new tools are the seeds of discovery and that basic science research is essential for the cures of tomorrow. As Dr. Beckman said, only by taking risks do you make progress, and we sincerely thank the Arnold and Mabel Beckman Foundation and the Beckman Young Investigator Award for allowing my lab to take major risks in developing Imagines and then following our discoveries where they take us. With that, I'd like to thank the people in the lab who did the work, our collaborators, and our funding sources, in particular the Arnold and Mabel Beckman Foundation. Thank you.